An insect bite. Once detected, it starts with something small, a wound you barely notice. But within hours, something is already inside, not feeding on what's dead, but feeding on what's still alive. This is not fiction. This is a real parasitic infestation known as Myiasis. And one of its most aggressive forms is caused by a fly called Cochliomyia hominivorax. The name itself reflects its biology. Hominivorax means eater of living tissue. Most flies are drawn to decay. They lay their eggs on dead material, where their larvae feed without resistance. But this species follows a different strategy. It requires living tissue to survive. The female fly is highly sensitive to chemical signals, substances released from even the smallest break in the skin. A minor cut, a surgical incision, an insect bite. Once detected, she lands and deposits clusters of eggs, often 100 to 400 at a time. Under warm conditions, those eggs hatch within 8 to 24 hours. The larvae emerge and begin to anchor themselves using specialized mouth hooks. From that point, they begin to move inward. Not randomly, but in a spiral, screw-like motion. This movement allows them to penetrate deeper into tissue, feeding continuously on living cells. This is not surface contamination, it is active tissue invasion. As the larvae grow, they release enzymes that break down surrounding tissue structure. This makes it easier for them to expand the wound. At the same time, the body responds. Inflammation increases, fluid accumulates. Bacterial infection may develop, and this creates a dangerous cycle. As the wound expands, it releases stronger odor signals. These signals attract additional flies. More flies lay more eggs. More larvae increase tissue destruction. This creates a self-amplifying process that can progress rapidly if not interrupted. Clinically, this may present as severe localized pain, visible movement within the wound, foul-smelling discharge, and rapid expansion of tissue damage. In advanced cases, larvae may penetrate deeper structures, including muscle. Historically, this parasite caused major economic and medical impact. Livestock industries were heavily affected. Animals developed infestations that reduced productivity, caused systemic illness, and often required euthanasia. Human cases, while less common, could become severe, especially when treatment was delayed. But one of the most remarkable solutions in applied science changed the course of this problem entirely. Instead of trying to eliminate the parasite by killing it directly, scientists targeted its ability to reproduce. This approach is known as the sterile insect technique. Male flies were bred in controlled environments, then sterilized, commonly using radiation. These sterile males were then released into the wild in large numbers. Female screwworm flies typically mate only once. If that mating occurs with a sterile male, no offspring are produced. Over time, this reduces the number of viable larvae in the next generation. As fewer offspring survive, the population begins to decline. With repeated releases, the reproduction rate approaches 0. And when reproduction stops, the population cannot sustain itself. Over multiple generations, the number of flies collapses. This method led to the elimination of the New World screwworm from the United States and much of Central America. It was not temporary control. It was a population-level eradication. However, eradication in one region does not mean global disappearance. The parasite still exists in parts of South America and certain tropical environments. Today, global conditions continue to change. International travel, animal transport, and environmental shifts all increase the risk of reintroduction. For that reason, surveillance systems remain active. Veterinary monitoring, Border inspections and rapid response protocols are used to detect and contain any new cases early. Because once established, this parasite spreads aggressively, especially in warm climates where its life cycle accelerates. From a clinical standpoint, management involves early recognition, mechanical removal of larvae, and proper wound care. Treatment of secondary infection may also be required. When identified early, outcomes are typically favorable, but delays can increase the risk of complications. This is why awareness matters. Not for alarm, but for understanding. Because this organism does not act randomly, it follows biological rules. It responds to chemical signals. And it depends on conditions that can be recognized and interrupted. The New World Screwworm represents two realities at once. It is a reminder of how aggressive natural systems can be. And it is also an example of how targeted scientific intervention can control even the most invasive threats. Some dangers are visible, others begin silently. And sometimes, the difference between the two is simply knowing what to look for before it becomes something more serious. Because sometimes what you don't see is exactly what you need to understand. Follow for more. Want to learn more? These trusted sources provide deeper insight into the science, spread, and control of this parasite. Because real understanding starts with reliable information. Sources are used for educational purposes only. No affiliation or endorsement is implied. This video is intended for educational and informational purposes only. It is designed to provide general scientific and public health information about New World Screwworm and related biological concepts. It is not intended as medical, veterinary, or professional advice, and should not be used as a substitute for consultation with qualified healthcare providers, veterinarians, or public health authorities. All information presented is based on publicly available sources believed to be accurate at the time of creation. However, scientific knowledge and public health guidance may evolve, and viewers are encouraged to consult official and up-to-date sources for the most current information. The organizations referenced in this video, including the Centers for Disease Control and Prevention, CDC, United States Department of Agriculture, USDA APHIS, International Atomic Energy Agency, IAEA, World Organization for Animal Health, WWOAH, and Merck Manual are cited strictly for educational and informational context. These organizations are not affiliated with, do not sponsor, and do not endorse this video or its content. This video does not provide diagnosis, treatment recommendations, or specific guidance for individual cases. If you believe you or an animal may be affected by any medical or veterinary condition, you should seek immediate professional evaluation from a qualified healthcare provider or licensed veterinarian. Some visual elements in this video may be computer-generated or artistically rendered for illustrative purposes. These visuals are intended to aid understanding and may not represent exact clinical or biological appearances. Viewer discretion is advised, as certain topics may be sensitive. The content is presented in a non-graphic, educational manner to promote awareness and understanding. By viewing this content, you acknowledge that the creator assumes no liability for any decisions made based on the information provided in this video.